Okay, well, thanks for the opportunity to give this talk. It's not a reason to panic, and it's certainly not a reason to give up using fungicides, but I think it is a wake-up call to ensure that cereal rusts are monitored for resistance and fungicide resistance management strategies are adopted in the control of this important set of pathogens. This is work done by Nicola Cook, a PhD student supervised by myself and Diane Saunders that are joining this, with co-supervision from Stefano Torriano at Syngenta. She took a genotypic approach to screen for fungicide resistance in necrotrophic cereal pathogens and applied the same methodology to a data set of stripe rust that had been accumulated in the Saunders lab. The take-home message are that we are seeing clear signs of evolving resistance to DMI and SDH fungicides in stripe rust. This confirms that cereal rusts are certainly not immune to fungicide resistance, as has been widely considered, and that I think that means we need to screen current isolates from all countries and establish a network of phenotypic efficacy controls, testing sites. So fungicides, I suspect, will remain a valuable component of cereal rust disease control and need to be used sensibly. Bit of background on fungicides, cereal rusts. Modern systemic fungicides were introduced into primarily Northwest Europe about 50 years ago and somewhat later in different parts of the world. They're used profitably pretty much wherever yields are over 2 tonnes per hectare. So that means that they're really widely used in not many areas of the world are not using fungicides. Different fungicide classes have been introduced at different times, so the MBCs are pretty much gone now. DMIs were introduced in the late 70s in the UK and perhaps 2005 in a serious way in Australia and other places. Rusts have been classified as low risk of fungicide resistance, whereas many other species develop fungicide resistance very quickly, powdery mildew, Botrytis septoria. The rusts didn't do so, and in fact there have been no cases of field resistance to a cereal rust fungicide. However, recently we've been seeing cases of the Asian soybean rust, Phacopsa pachyriza, developing resistance to QOI, DMI and SDH fungicides in South America. So this means that we have to consider the possibility that this is also happening in the cereal rusts. There are 4 main classes of fungicide that are used to control cereal rusts around the world: DMIs, SDHIs, strobilurins and multisite inhibitors. And there are 3 main types of fungicide resistance mechanism. There are target site mutations, where the protein sequence that is targeted by the fungicide alters its structure so that it binds the fungicide less tightly. There's target site overexpression, where normally insertions in the promoter of the target site gene lead to overexpression, so you simply have more of the fungicide target, so you need more fungicide to receive the same control effect. Or efflux bumps, where the fungicide that has managed to get inside the fungal cell is pumped out. These 3 target site mechanisms, fungicide resistance mechanisms, tend to occur in order, with target site mutations first and the other 2 following on as the time passes and selection pressure increases. In these 4 fungicide classes that we're thinking about, multisites haven't really had any fungicide resistance associated with them, and we weren't able to look at the QOI fungicides for a technical but soluble reason. In the case of the DMI and the SDHI fungicides, there are well-known target site mutations that are essentially the same amino acid in different species. In the case of DMIs, it's the CYP51 gene or genes that is affected, and many different fungicide resistance-associated mutations occur, and they occur in many, many different combinations. Each combination has a different impact on a different DMI fungicide. In the case of SDHIs, we tend to see single mutations in one of the 3 genes. These have relatively high resistance factors and affect the different SDHI fungicides to a different extent. So what we did in this study, what Nicola did in this study, is she took the genomes of 363 isolates of stripe rust from 24 countries: UK, Europe, New Zealand, China, USA, Pakistan, and Ethiopia. They were collected from about 1989 to 2016. And she screened them for mutations in the sequenced SDH, B, C, and D genes and the CYP51 genes. And the result was she found 12 isolates with a relevant mutation in the SDHC subunit I1I85V, and 26 isolates with this mutation in CYP51Y134F. And you can see both mutations were found in New Zealand. Looking at these mutations in more detail, the CYP51134F mutation was found in half the isolates from New Zealand and 2/3 of the isolates from China. This mutation is equivalent to Y137F, which has been found in many other species. It's associated with weak resistance to the older DMA fungicides such as triadimenol, tebuconazole, propiconazole, and is a gateway mutation. So those early DMIs select for, first of all, this mutation and then second mutations. And in the case of barley powdery meal, for example, we saw Y134F plus another mutation lead to $50 million a year losses in West Australia from about 2010 for a few years. In the case of this stripe rust, the collections were made when early DMIs were still in use in China and New Zealand. My expectation is that the current isolates would not be showing much in the case of phenotypic impact, particularly when the fungicides that are being used in China and New Zealand, which are based around prothio and epoxiconazole, are being used. In the case of the SDHI mutation, C185V, this was found in 2 isolates from each of UK, Ethiopia, New Zealand, and all 6 from Pakistan. It's equivalent to a site in the P-Teres archetype SDHC P73. Mutations at P73 or its equivalent amino acids have been found in 4 other species, interestingly including the Asian soybean rust pathogen. These mutations are associated with medium level resistance factors about 20, so I would guess that they would show phenotypic alterations consistent with field failure if the concentration of these isolates was high in the affected regions. So what's the implications of these findings? So what we found is that random samples from diverse growing conditions, low, medium and high yield conditions with low, medium and high fungicide intensities, are showing high frequencies of resistance associated genotypes. What this does is it increases pressures on genetic resistance, because we know that genetic resistance protects fungicide sensitivity and vice versa. And I think what this says above all is that we need to ramp up our monitoring for resistance to fungicides in the cereal rusts, all 3. Both genotypically, because that's much easier technically. We need to get isolates from all relevant countries. We need isolates that are current, and we need to look at all the relevant target genes and promoters. Phenotypically, it's also important to do that as well. It's technically more complicated, but gradient bait assay is a good way to start because it selects for the isolates that grow on the highest fungicide doses. Overall, though, we need to implement already, I think, for cereal rust, resistance management strategies that we use for protecting fungicide sensitivity and other pathogens. And these can be classified into both the chemical and non-chemical resistance management strategies. So in the case of chemicals, that means mixing the fungicide mode of actions we use, limiting the number of applications in a season, using the minimum dose that gives an economic return, and spraying early to stop there being a large amount of inoculum. Non-chemically, first foremost, is to use genetically resistant cultivars that have stable and broad spectrum resistance. But then we can throw in the agronomy like crop rotation, farm hygiene, stubble management, green bridge destruction, and alternate host control. Overall, the take-home message is don't be too keen to be too clean. Otherwise, the selection pressure for breaking down either the chemical or the genetics is overly high. So I just want to thank the people who contributed to these studies, people that joined us with and Indian Curtin who helped with technical aspects, and our friends Stefano and Susan Knighton-Syngenta who provided information and discussions, and I'll be very happy to answer any questions on this. Thanks very much for your attention.